Hello. We're going to continue making these short videos on different subjects in my microbiology class. This time we're going to be talking about the control of microbes. This is basically going to go through the history of sanitation, the different types of antibacterials, antimicrobials that we have, and different methods into applying them. So, to start off, we can't really talk about all that cleanliness and the process of making things septic or disinfected without talking about Joseph Lister. He lived in the 1800s, so from the period of 1827 to 1912, and he's known as the father of modern surgery. It is said that he saved more lives in his system of creating ways to prevent infection than all of the lives that were lost in the 19th century. So he was able to revolutionize surgery. By lowering infections with his methods. One of his methods would be the usage of carbolic acid, which is a phenol. This chemical, let me change to a different highlighter. This chemical would be sprayed in rooms as well as tools for surgery at the time. And since we're talking about the 19th century, it's very crude. Back then, they just had saws. There isn't many high-tech things as you would think today. A lot of things weren't sterile. So by introducing this, he was able to really decrease those infections. They're also put on surgeons' hands. He then began to sterilize instruments, as well as a clean operating environment. So this is where we're going to see more attention being put into the fact that we need to prevent infections when we're doing operations in medicine. And overall, we're going to see this transition into the study of diseases, as well as, of course, bacteriology, and then a little bit of virology as well. There is a challenge when killing microbes, so the challenges that we'll come across stem from the types of microbes. So you have vegetative cells. Vegetative refers to the cells that are metabolically active, and therefore these cells were actually going to be able to divide. On the flip side, we also have much hardier cells or much hardier microbes, and specifically have these in endospores. There are a lot of other types which we'll get to in a bit, but the most general will be the bacterial endospores, and these are inactive. They're very resistant and they're dormant. They survive tough conditions. And these are much harder to kill than vegetative cells, as they are hardy. They're able to remain dormant in conditions that may usually kill bacteria. Alright, let's talk about the principles of control. So before we get into the methods, we need to know some definitions. So bactericide, this means it will kill bacteria. Bacteriostatic, key word here is static. So side Again, that means kill, so a bactericide kills bacteria, while a bacteriostatic will just slow them down in their growth. It doesn't kill the bacteria, it just inhibits growth. More definitions in describing how clean or how void of microbes something is. We have sterilization. I'm going to change the color. So sterilization is when we do not have any microorganisms at all. So this includes no, when we say no microbes, this will include no bacteria, viruses, but prions, those misfolded infectious proteins, these are still going to be present. Prions are extremely difficult to get rid of, and one of the techniques would be raising the temperature to such a high degree which will potentially end the prions, but that is a very complicated process. It takes a lot of energy input to get rid of prions. So sterilization gets rid of everything except for prions because they're just so hard to get rid of. Disinfection. This means the elimination of most or all pathogens. Again, a pathogen is something that causes disease, and some viable microbes will remain. So sterilization, you're getting rid of everything. Disinfection, some of those viable microbes will still be there. Disinfectants, so when we have disinfectants, this means chemicals or methods used on inanimate objects. So if I have a bench outside and I spray a chemical on it, since that bench isn't alive, it's inanimate, that would be called a disinfectant. For living tissue, we actually will be using antiseptics. This is, again, going to still be the same type of thing. We're using a chemical to get rid of these microbes, but this is only going to be for living tissues. Alrighty. Kind of like an alcohol wipe before getting a flu shot or something like that. First person to use this term, antiseptic, is actually an ancestor of one of my professors, John Pringle, and he was a physician in Scotland from the 1700s. He really worked with military medicine, and so again he was able to really create antiseptic techniques and advance them for the purpose of the Scottish Army, and so a lot of people are able to get healed thanks to his work. That's a little bit of an aside. There isn't that icon that is needed for like memorization on that slide, but it's just a little fun fact we like to throw in there. Alright, more definitions, it looks like. So, we also have to talk about decontamination. This is the reduction of pathogens to safe levels. So they're still there. They're just going to be at safe levels. The pathogens are still present on whatever surface that is. They're just down to safe levels. We don't have to worry about an abundance of them. Sanitization is when we reduce the pathogens to a significant amount that meets the health standards. So there's a difference, again, reducing the amount just to safe level and then significantly reducing them to whatever health standards are in place, probably by the CDC or another organization like that. But I think what's trying to be pointed out is sanitization is stronger than decontamination. We have pasteurization, again from Louis Pasteur, which we've covered a bit on this channel. This is a brief heating to reduce the number of spoilage organisms. We also destroy pathogens here. Then we have one more term on this specific slide. We also have preservation. And this is the delaying of spoilage. We aren't killing anything in preservation, however, we're just delaying the growth or inhibiting the growth of those microbes on perishable products. Alright, in labs, this is another aside, so I'm just going to go through it briefly. So in labs, the control of microorganisms includes the use of control methods, and sometimes there are specific control methods in place. So you'll have actually different safety levels, known as biosafety levels or BSLs. You have 1 through 4, 4 being the most strict when you're dealing with very terrible and dangerous high-level pathogens. BSL 1, however, it's kind of pretty tame. That's the tamest and least concerning biosafety level. So 4 is the highest, 1 is the least troubling. So BSL 1 through 4. We need to eliminate contamination from samples and surroundings. This includes aseptic technique, which we've covered before as well. Alright, and usually you want sterile tools as well as cultures. Alright, that's going to again be an aside, so it's not the main focus of all these lecture slides, but the professor went over it, so might as well include it for a bit. Alright, so now in manufacturing, this is again an aside, so food production, tons of regulation in food production. The canning process, again, we talked about pasteurization. You could also have irradiation. As well as chemical additives. And facilities must be kept clean and organized, of course. Other things that may contain microbes, such as cosmetics, are also regularly screened for any microbial contamination. So we have a lot of regulations in place in food production. Hopefully we get some of those regulations updated, because again, you don't know what types of microbes are out there, so we just need to make sure we take precaution when we're manufacturing or preparing food always. Alright, control and water treatment. Let's switch to blue because we're going to be talking about water now. So, we need to make sure drinking water doesn't have any pathogens. Of course, that's the main goal. So, usually we'll use chlorine. And unfortunately, the chlorine can react with natural chemicals in the water to create disinfection byproducts, DBPs, and these have health risks. So we have to monitor the amount of chlorine. Even though it does work in disinfecting water for the most part, we have to monitor it because of these DBPs. Another thing we need to look at are some resistant organisms, such as this bacteria. I'm not going to pronounce it because I'm just going to fumble it, but Cryptosporidium, Cryptosporidium, and then parvum, I think. And this is the bacteria that causes diarrhea, so we, of course, want to prevent that as much as possible. We need to look at other treatment methods, because if it's resistant to chlorine, that's a big problem. So regulations require facilities to minimize DPPs and the diarrhea bacteria, so parvum in H2O. So again, regulations highly important here, making sure everything is safe. So what affects the antimicrobial procedure? Many, again, of the microbes will be resistant. So again, we talked about endospores. These are the most resistant. And again, they are inactive, and they tolerate great heat. These are extremely dangerous. There's, of course, the stories of like anthrax exposure, because anthrax makes endospores, and the toxins that the bacteria anthrax causes, it is extremely deadly and extremely serious to deal with. That's why there's always such a big worry and scare when we're talking about endospores, because of species such as anthrax. We also have protozoa, and these make cysts as well as oocysts. I do not know how to pronounce that, so I'm just going to say oocyst. These are resistant to disinfectants. Remember, we looked at the difference between a disinfectant and a sterilant. So disinfection is getting rid of most pathogens. Again, some viable microbes will remain. A sterilant gets rid of all. So these are going to be resistant to disinfectants. These are found in feces, and they also cause diarrhea. There's 2 R's. Yeah, sorry about that. I had a brain fart. Okay, so Mycobacterium. Again, the Mycobacterium, they do not have a cell wall that has peptidoglycan. We have a waxy cell wall in these. So that makes them resistant to substances like penicillin. Because penicillin will attack the peptidoglycan, it will affect the cross-links in that substance in the cell wall. But since Mycobacterium, they do not have that, they have a waxy cell wall, they are resistant to chemicals such as penicillin. We also have Pseudomonas. We've dealt with this bacteria recently in the lab portion of class. They are resistant to, as well as they could also grow in some disinfectants. So they're kind of like the diarrhea bacteria, where it can grow in chlorine, but the Pseudomonas kind of have the same thing. We also have a danger from non-enveloped viruses. So something like COVID-19, that of course has a protein coat, that's envelope, but we're talking about viruses that do not have that lipid envelope. And these are, again, resistant to disinfectants. All right, let's go into more factors. So let's talk about the number of microbes. This is another factor that will control the procedures, how they're going to be dealt with. So the number of microbes determines the time for heat or the amount of chemicals. And a fraction of the population will die during the given time interval. So a large population, so of course if we have a ton of bacteria, sorry about that, a brain fart, that means more time will be needed. And removing organisms by washing will reduce this time that is needed. Okay, so for example, we have the decimal reducing time, or reduction time. And this is how we talk about the effectiveness for commercial reasons. So the time required to kill 90%. 90% of a population under specific conditions. This is referred to as a one-log reduction. And there's a chart here in this slide which I can't really draw in great detail, but there's 10,000 over here. This is going to be the number of surviving cells. We have 1,100. Let me make 100 a little bit lower. Then 10. And then, of course, 1 will be our bottom, it looks like. And then over here we have 0 to 5, 10, 15, 20. And that's the end of it. Okay, so when we have 1,000 surviving cells, that's going to go to 5 minutes of time at the given temperature. If we have, again, if we have 10 remaining cells, that will be also 5 here, which is very interesting. There's going to be a line going down as we go, but if we set up the grid even more, so if we go like that, and this line will go from the top here, so 10,000 surviving cells, to diagonal line all the way to the bottom. And this time here, so if we had 10 surviving cells, this is called D. This is our decimal reduction time. I'm sure that he'll go into more detail about what all of that is. Let me look what we talked about in class here. It's basically the same thing. Those were my notes from class. I'm going to have to ask the professor about that because I really do not know how to explain it. Oh well, moving on. Sorry about that. That was really horrible for this video to go into that rabbit hole without any success. Oh well, let's just keep moving on. Heat treatment is also used for microbial control. It's safe. We know it works, so it's reliable. It's pretty fast too. And also, the biggest one, it's probably non-toxic. You're not using any weird chemicals. You're just using good old heat. You could use heat for sterilization or disinfection. Alright, we actually have two types of heat when we're using it to destroy microorganisms. We have moist heat. And I write a big word there, moist, in big font. And this will denature proteins. This includes boiling, which will destroy most microbes, as well as some viruses. Endospores will survive though. So again, they're very hardy, so these actually survive. Pasteurization is another type of moist heat. This is the brief heating to destroy heat-sensitive pathogens, as well as spoilage organisms. So spoilers, I guess you could just call them spoilers. This is of course used for milk, beer, straw milk carton. You know, recently on the news in the US there's that, have you seen that fighter jet thing? So, you know, in the side of a milk carton sometimes you'll see that, maybe. Have you seen the fighter jet? You know where it is? We also get it, pasteurization in beer, so maybe a can of Bud Light. There's a can of Bud Light. As well as juices, so V8. There's high temperature short time, so HTST, high temperature short time. This is used for most products. This is going to be in milk, which is 72 degrees Celsius for 15 seconds. There's also ultra high temperature time, so UHT. This is going to be 134 degrees Celsius for 2 seconds. And this is going to be used for boxed juice, because you're going to just leave it on there on the shelf for much longer. So we need a little bit of a higher heat. We need it to be shelf stable. That's what we're going for when we use UHT. We also have steam techniques. So one of these methods is the autoclave. And again, we're going to be using steam and pressure to sterilize. That means, again, kill all the microbes. So the increased pressure will raise the temperature, and this will kill endospores. The specific temperature that's usually is 121 degrees Celsius at 15 psi. That's again your pressure units in 15 minutes. And of course you'll increase this if needed for longer values and larger volumes. There's also flash sterilization, which is, again, higher temperatures, so we're going to increase even past 121 and then even quicker. Prions, those prions are prions, which we were discussing earlier. These ones can't be killed with sterilization, but we could get to high enough temperatures to actually knock them out using the autoclave. So that's going to be 132 degrees Celsius for an hour. That is going to be a much more, much more energy consuming, as well as, look at that, 132 degrees Celsius for an hour. That is an extremely harsh condition, so that's going to be able to get rid of those prions, those infectious proteins. Alright, another aside, this one's actually fun. So Napoleon, from French history, Napoleon Bonaparte. So the French government offered a prize for a method of preserving food. So in 1810, a guy named Appert, he had this award of 12k francs because he found out that heated food in boiling water with glass jars with a cork, this would actually preserve the food. And then also around at the same time in the same year, there's a guy named Durand who uses the patent of the tin can for the first time. So again, Napoleon fought a lot of wars and all these battles and all these crazy things. If you read all about French history, he did a lot of interesting stuff and a lot of crazy stuff, but he needed something to feed the army, of course, and if the army's traveling everywhere, they needed a method to preserve food. So he awarded a pot for the invention of finding a way to preserve that food. It was kind of like a challenge, who could preserve the food the best. So what he did is put the food in some boiling water. Here's the water, here's the food, and he sealed it, and so that was able to preserve the food until it was shipped off somewhere else, and then the soldiers could eat it. There was less spoilage. Little interesting tangent for you. Again, this goes to the canning process. So the canning process actually uses a giant autoclave called a retort, and we want to again destroy the Clostridium botulinum. If you have pink sauce, unfortunately, those aren't, autoclaved in any way, so you're going to probably get Botox poison from eating that pink sauce that is just left on the shelf and all those raw ingredients. So don't do that. Be cautious of what you're eating. It will reduce the endospores, so 10 to the 12th endospores will be reduced to only 1. And this is critical because endospores, again, germinate and then they create the toxin. So we're reducing all those endospores, 10 to 12, that's a huge number. We're just reducing it to 1. That's a 12D process. And canned food is commercially sterile. It has to be sterile. However, endospores of thermophiles may survive. So just again, food for thought, no pun intended. Please don't eat pink sauce or anything that contains raw food or ingredients in an unautoclaved or unsterile process, because that's really bad. You don't want those Clostridium botulinum ruining your day. Alright, dry heat. Let's talk about dry heat. We already talked about moist heat. Let's talk about dry heat. This is less effective because of longer time and higher temperature. So again, more energy is spent. For example, it's going to be 2000 degrees Celsius for 90 minutes versus the moist heat, which is only 121 degrees Celsius for 15. I'd rather do the 121 degrees Celsius for 15 because of the lower heat that I need to get to, as well as the time. It's much quicker. So hot air oxidizes cell components, and it also, of course, denatures. So the proteins will also be attacked here too. This is going to be for glass, powders, and oils. A lot of dry material too. Incineration, so this is a form of it. So in incineration, you're going to turn the cells to ash. Like ash ketchum, but, like volcanic ash, so like burnt up ash, not ash ketchum. Used to destroy medical waste. So again, in lab, we'll have these inoculating loops, and we're going to use the Bunsen burner. That is a form of incineration, but you'll see medical waste be destroyed this way too. We also get rid of carcasses. in the medicine as well as in the biological field, you get rid of carcasses through incineration. And we mentioned already the loop and the Bunsen burner is a method used in schools or in colleges when they're teaching labs. So you, of course, flame the loop, and that is incineration. Other physical methods, we also have filtration, either of fluids or the air. First, we're going to focus on fluids. And you're going to use this for heat-sensitive materials because you don't want to be burning something that will just set the whole place on fire or anything like that. You want to try and be smart and find other physical methods in order to treat these things. So membrane filters. We have pores that are about 0.2 UM, extremely tiny, and this is going to remove bacteria. We also have depth filters. These are going to be thicker with larger pores, and then we're going to use electricity to trap cells. Right, filtration of air. Let me just highlight the things that are important here. So air, one of the four elements. How are we going to filter it? So we're going to be using high efficiency particulate air filters, HEPA. And these are going to remove almost all microbes. We could also use radiation, different types of radiation like UV, gamma rays, X-rays, so let's discuss those. And this removes electrons from atoms. Again, gamma rays like the Hulk, or X-rays are used, and these destroy DNA. They damage the cytoplasmic membranes, and they react with O2 to form those reactive oxygen species, which are extremely dangerous. Again, this is for sterilization. And the FDA allows radiation for insect control. We also use it for parasites, in pork specifically, as well as bacterial control. And you'll see the insect control done on spices and herbs. You'll see bacteria control done on different types of meat. We also have UV radiation. This one is a little bit different. It's on its own slide, so let's go through it. So this is direct destruction of the microbes. Again, damages DNA, and it could go on air, water, or surfaces. However, it can't really penetrate. It doesn't go that deep. If there are thin films or coverings, these will limit its effectiveness, nor can it kill microbes who are in solids or turbid liquids. Microwaves. Also are used to kill by radiation. These are going to be heat-based. And microwave ovens are going to heat food unevenly, so some cells are going to survive. And I should note that this is uneven. Alrighty, let's go to our next part. In the next video, we'll be going over chemical methods used to destroy microbes. Thanks so much for tuning in, and please do something nice for someone.